Good morning everyone. I am Dr. Shashi Rekha. Today's physiology class is Cerebellum 1. Contents: Functional anatomy of cerebellum, physiological divisions, deep cerebellar nuclei, afferent and efferent pathways, neural circuit, functions of each part of cerebellum. Introduction. Largest part of hindbrain controls ipsilateral side of body. Location. Occupies most of posterior cranial fossa. Lies behind pons and medulla. Forming roof of fourth ventricle. Separated from posterior part of cerebrum by tentorium cerebelli. This diagram is showing cerebellum and its location. Anatomical divisions of cerebellum. Anatomically, it is divided into anterior lobe, posterior lobe, flocculonodular lobe by primary fissure, posterolateral fissure or uvulonodular fissure. This diagram is showing anterior lobe, posterior lobe and flocculonodular lobe. Physiological divisions. There are three physiological divisions. Number 1, vestibulocerebellum, also known as flocculonodular lobe. Second, spinocerebellum is formed by vermis and intermediate zone. Number 3, cerebrocerebellum is lateral zone. Functional divisions. Functionally, the divisions or lobes are named according to the connections they make with other components of the motor control system. The flocculonodular lobe, it is functionally related to the vestibular apparatus and therefore also called the vestibulocerebellum. It is concerned with control of body posture, equilibrium and maintaining visual fixation, that is vestibulo-ocular reflex. The entire anterior lobe and parts of the posterior lobe, lobulus simplex, pyramis, uvula, parafloccule, that receive information from the spinal cord are called the spinocerebellum. It occupies the median portion of the cerebellar cortex and receives proprioceptive input from the body. It is concerned with the control of axial, that is trunk, and limb muscles and postural reflexes. The remaining part of the posterior lobe receives information from the cerebral cortex and pons and thus called the neocerebellum. It occupies the more lateral regions of the cerebellar cortex. It is concerned with skilled voluntary movements. Cerebellum has two main gray structures. A. Laminated cerebellar cortex, cerebrocerebellum, spinocerebellum, vestibulocerebellum. B. Deep cerebellar nuclei, dentate nucleus, globus and emboliform nucleus, fastigial nucleus. Peduncles and deep cerebellar nuclei are the inputs and outputs of cerebellar cortex. This diagram is showing superior, middle, inferior cerebellar peduncles and fastigial nucleus, interposed nuclei, dentate nucleus, deep cerebellar nuclei. All these are deep cerebellar nuclei. Cerebellar cortical neurons. Before going into the cerebellar cortical neurons, we will discuss cerebellar cortical gross structure. Grossly in cross section, the cerebellum is seen to have an outer grey matter called cerebellar cortex that surrounds the inner white matter. Within white matter are embedded 4 pairs of nuclei. The deep cerebellar nuclei. The cerebellar cortex is extensively folded on itself, constituting the folia, that is leaf-like parts which are marked off from one another by fissures. These folia overlie the white matter, which consists of afferent and efferent fibers. Now coming to the cerebellar cortical neurons. Microscopically composed of 3 cell layers. Number 1, the innermost layer is called the granular cell layer. Second, the middle layer is the Purkinje cell layer. Third, the most superficial layer is called the molecular layer, which is almost devoid of cells. 5 types of neurons are present. Number 1, basket cells, and second, stellate cells, that is molecular cell layer. Third, Purkinje cells, that is Purkinje cell layer. Fourth, granule cells, glutamate, that is glutamate. And fifth, Golgi cells, granular cell layer. Pause. Layers of cerebral cortex. See the diagram. It contains endings of unmyelinated nerve fibers and nerve cells. Number 1, the nerve endings are: number 1, the dendrites of the Purkinje cells. Number 2, the axons of the granular cells. And number 3, the afferent fibers from inferior olivary nucleus, which ascend into this layer as the climbing fibers and form synapses with dendrites of the Purkinje cells. Second, the nerve cells are of 2 types. The star-shaped stellate cells, more superficial in location, and second, deep located basket cells. These cells possesses a relatively sparse dendritic tree which receives input from the parallel fibers. They are so called because their axons from a basket around the cell body of each Purkinje cell they innervate. Purkinje cell layer. It is only one cell thick, consisting of large flask-shaped Purkinje cells, the biggest neuron in the body. These are about 15 million Purkinje cells in man. Their axons from the only output of the whole cerebellar cortex, which passes to form synaptic connections in the deep cerebellar nuclei. They have very extensive dendritic network that extends vertically into the outer molecular layer and provides a huge surface area for axodendritic synapses. Next, granular cell layer. The main cellular component of this layer are granule and Golgi cells. Number 1, granule cells. They are small, very numerous, about 10 billion, and have their cell bodies located in this layer. Each cell axon ascends to the outer molecular layer and then bifurcates to form a T. The 2 branches of the T run along the long axis of the folium and are called parallel fibers. The parallel fibers make excitatory synaptic contact with dendrites of many Purkinje cells, Golgi cells, basket cells, and stellate cells. Golgi cells. These are large cells and less numerous than granule cells. Their dendrites project outwards into the molecular layer and receive input from the parallel fibers. Their cell bodies receive input via collaterals from the incoming climbing fibers and Purkinje cells. Their axons branch extensively and form inhibitory synaptic connections with the dendrites of the granule cells. Afferent and efferent pathways. Input signals. Number 1 from respective body parts. Number 2 corresponding topographical areas of motor cortex. Output signals. Same areas of motor cortex. Second, red nucleus. Third, reticular formation. Principal afferent tracts to the cerebellum. Superior cerebellar peduncle from midbrain, middle cerebellar peduncle from pons, inferior cerebellar peduncle from medulla. This diagram is showing peduncles, lobes, afferent tracts to the cerebellum. The peduncles are superior cerebellar peduncle, middle cerebellar peduncle, inferior cerebellar peduncle. The lobes are anterior lobe, posterior lobe, flocculonodular lobe. Tracts are ventral spinocerebellar tract, cerebropontine tract, postcerebellar tract, vestibulocerebellar tract, olivocerebellar and reticulocerebellar tract, ventral spinocerebellar tract, dorsal spinocerebellar tracts. Inputs from cerebrum. Temporal and occipital cortices provide the cerebellar cortex with signals associated with auditory and visual functions. Somatosensory signals also reach the cerebellar cortex from the cerebral cortex. The desynaptic pathway from the cerebral cortex to the cerebellar cortex with a synapse in the deep pontine nuclei. Cortico-pontine fibers are shown in red and pontocerebellar fibers are shown in blue. See this diagram. In this diagram, cortico-pontine fibers are shown in red and pontocerebellar fibers are shown in blue. Input from brainstem. These include pathways arising from the red nucleus shown in blue, deep pontine nuclei shown in red, vestibular nuclei and reticular formation. In humans, the inferior olivary nucleus appears to transmit an integrated signals from the spinal cord and cerebral cortex to the cerebellar hemispheres via the climbing fiber system. In this diagram, pathways arising from red nucleus shown in blue, pathways arising from deep pontine nuclei shown in red. Next, input from spinal cord. The pathways most critical for supplying such inputs to the cerebellum include A. The dorsal spinocerebellar tract, B. Ventral spinocerebellar tract, C. Cuneo-cerebellar tract and D. The rostral spinocerebellar tract. Fibers from the spinal cord convey information from cutaneous afferents, joint afferents and muscle spindle to the inferior olivary nucleus. From this structure, axons pass to the contralateral cerebellar cortex via the inferior cerebellar peduncle, terminating somatotropically within the anterior and posterior lobes of the cerebellar cortex. Therefore, these receiving areas are referred to as the spinocerebellum. The diagram here showing A. The dorsal spinocerebellar tract, B. Ventral spinocerebellar tract, C. Cuneo-cerebellar tract, D. The rostral spinocerebellar tract. Input to the cerebellar cortex. Number 1. Climbing fibers originate from a single region of the medulla, inferior olive, make multiple synapses onto Purkinje cells, resulting in high frequency bursts or complex spikes. Condition: the Purkinje cells play a role in cerebellar motor learning. You can see all these things in the diagram. All the afferent fibers enter the cerebellum through two main sources of inputs. A. Climbing fibers. They come from the inferior olive. They give off excitatory collaterals to the deep cerebellar nuclei and terminate on dendrites of Purkinje cells. Second, Morse fibers originate from many centers in the brainstem and spinal cord. Include vestibulocerebellar, spinocerebellar and pontocerebellar afferents. Make multiple synapses on Purkinje fibers via interneurons. Synapses on Purkinje cells result in simple spikes. Synapse on granule cells in glomeruli. The axons of granule cells bifurcate and give rise to parallel fibers. The parallel fibers excite multiple Purkinje cells as well as inhibitory interneurons. That is basket stellate Golgi type 2. Output pathways from cerebellum. All output signal originate or pass through in deep nuclei, that is brain. Three major output pathways. The diagram is showing cerebello-thalamo-cortical tract to thalamus, red nucleus, reticulum of mesencephalon, superior cerebellar peduncle, fastigioreticular tract, fastigial nucleus, fastigioreticular tract, paleocerebellum. Major cell types. Granule cells secrete glutamate. Purkinje cells secrete GABA, that is gamma amino benzoic acid. Golgi cells secrete GABA. Stellate cells also secrete GABA. Basket cells secrete GABA. The Purkinje cells is the only output of the cerebellar cortex. Purkinje cells and deep nuclear cells fire continuously. Afferents excite the deep nuclear cells. Purkinje cells inhibit the deep nuclear cells. Main connections of the vestibulocerebellum. Input and output of the cerebral cortex. Input: climbing fibers plus excitatory from inferior olive nucleus. Morse fibers plus excitatory form spinal cord and brainstem. Output: Purkinje cell axon minus inhibitory. Components and connections of functional divisions of cerebellum. Division: vestibulocerebellum. Components: flocculonodular lobe. Afferent connections: vestibulocerebellar tract. Efferent connections: cerebellovestibular tract and fastigiobulbar tract. Division: spinocerebellum. Flocculonodular lobe. Components: spinocerebellum. Components: tuber, pyramid, uvula, parafolliculi and medial portions of cerebral hemispheres. Afferent connections: dorsal spinocerebellar tract, ventral spinocerebellar tract, cutaneocerebellar tract, olivocerebellar tract, pontocerebellar tract, tectocerebellar tract, trigeminocerebellar tract. Efferent connections: Fasciculobulbar tract, cerebello-reticular tract, cerebello-olivary tract. Division: corticocerebellum. Components: lateral portions of cerebral hemispheres. Afferent connections. Pontocerebellar tract, olivocerebellar tract. Efferent connections: dentatothalamic tract, dentatorubral tract. These are the components and connections of functional divisions of cerebellum. Coming to the output pathways from the cerebellum. Efferent fibers from the cerebellum are axons of the deep nuclei. 1. From the vestibulocerebellum. Fibers originate in the vermis and pass from the fastigial nuclei to reticular formation and to the vestibular nuclei, which then give origin to reticulospinal and vestibulospinal tracts. Thus, this part of concerned with the control of equilibrium, as it helps the control of movements in axial, neck, shoulder and hip muscles. 2. From the spinocerebellum. Fibers pass from the nucleus interpositus located in the medial part of the cerebellar hemispheres to A. The ventrolateral and ventroanterior nuclei of the thalamus and then to the motor area of the cerebral cortex. B. The medial thalamic nuclei and then to the basal ganglia. C. The red nucleus and reticular formation. This circuit is important in the coordination of movements in muscles of the peripheral part of limbs. 3. From the cerebrocerebellum. A pathway from the lateral zone of cerebellar hemisphere pass to the dentate nucleus to the ventrolateral and ventroanterior nuclei of the thalamus and finally to the cerebral cortex. This part's pathway is important in coordinating and planning sequential movements. The cerebellar cortex has 4 major efferents. Purkinje cells are the only output and are always inhibitory via the neurotransmitter gamma aminobutyric acid. B. The spinocerebellum has efferents to the red nucleus and reticular formation, which influences lower motor neurons via the reticulospinal and rubrospinal tracts to adjacent posture and affect movement. C. The pontocerebellum has efferents first to the thalamus and then to the cortex and influences lower motor neuron via the corticospinal tract. These efferents produce precise sequential voluntary movements. The vestibulocerebellum has efferents to the vestibular nucleus and elicits positional changes of the eyes and trunk in response to head movements. This is the end of the class. The remaining portion will be continued in the next class. Thank you.